Thank you very much, Gordon. It's a pleasure to, as always, to be in Split and to participate in this exciting meeting. So, going back to cancer, and before that, I would like to just, well, after the very nice talks that we had yesterday and today, just to remind you that when we talk about glycosylation in any eukaryotic cell, we have to think not only on O glycans, protein O glycosylation and N glycosylation, but also on many other types of glycoconjugates that I just wanted to make this point here, because obviously there's a lot to be understood also on those glycosphingolipids and these glycosaminoglycans. And after the very nice presentations, also I don't need to recall you about the very nice organized mechanism of protein N glycosylation, in which there are several steps codified by different genes that are crucial for these initial steps. And then when you move on to the later steps, you have families of glycosyl transferases, like cellular transferases, fucosyl transferases, and it was nice to see that some of the data that shows when you change one, you may have in these families others that could compensate or could interfere with the glycosylation. In the protein O glycosylation is quite different in some of the initial steps. So, for example, just the first step in protein O GalNAc glycosylation is mediated by a family of 20 different polypeptide GalNAc transferases, so 20 genes available. Of course, they have cell and tissue specificity and tissue expression specificity. And of course, you have in normal cells, these O glycans are extended, some branches making the different cores and extended. And the terminal structures, as we always see, different fucosyl transferases and sialyl transferases that will lead, for example, for the biosynthesis, control way of biosynthesis of the Lewis antigens, for example. And this is highly controlled in normal physiological conditions. This is just an example in the gastrointestinal tract, in the normal gastric mucosa. So the differentiation program towards the apical part of the mucosa leads to the biosynthesis of H-type 1, Lewis A, and Lewis B, whereas in the deep glands of the mucosa you have type 2 Lewis antigens, Lewis X and Lewis Y. And this is highly controlled, so following the program of differentiation of the cell. Well, so as I mentioned, and also nice to hear even today that if you do a completely unbiased search, you find that some other key markers, and yesterday, like key genes controlling this leading to this glycosylation features, the glycophenotype. Of course, glycosyl transferases, as expected, but also there are many other factors, like we have heard that molecular chaperones, transporters in the ER, in the Golgi, and ion transporters, and of course also substrates. Like unbiased search identify some substrate proteins being responsible, also interfering with this glycosylation phenotype. And in cancer, there has been a lot of data demonstrating with different levels of importance, of course, which includes mutations, epigenetic alterations, but also metabolic issues. And we cannot forget, of course, in the context of the tumor microenvironment, the inflammation and the immune response. And this ends up, as Pauline was showing very nicely yesterday, that some glycans have been particularly identified in the cancer context and very well documented in different studies, but also having a crucial function in the biology, in the tumor biology setting. And glycosylation, cancer is not a glycosylation disease, like Manfred was commenting yesterday. It's true, it's not a glycosylation disease, but there have been very well documented steps in which glycans play crucial roles in different different mechanisms, like invasion processes, interaction with extracellular matrix, of course, signaling mediated by receptors, and as Pauline was also mentioning yesterday, the process of extravasation and metastasization to the distant site of metastasis. Very well documented studies on that. So today I don't have very much long time, but I just wanted to, I selected a couple of topics. One, I just introduced a few slides because some people asked me to include that part. So I'm presenting some data on the biomarker, or an example of a biomarker that I think we could in the future introduce in the clinical precision nutrition, precision medicine setting. In fact, for example, in most, in the clinical setting, in tumor, in the oncology setting, a lot of the guidelines include the in-depth characterization of the tumor, and in most of the tumor you can actually identify molecular subtypes. And for this is an example in the stomach cancer example, in which there are clear different biological entities with different etiologies and with different key genes codifying key proteins in the oncogenic processes. And one of these subtype are enriched with receptor tyrosine kinases, and actually in the clinics is the guidelines include the use of drugs, in this case monoclonal antibodies that target the receptor RB2 in a particular setting of the advanced stomach cancer. And there are a couple of antibodies, and including trastuzumab. So I included that because in a study that we did a few years ago, Enrique Duarte, in collaboration with Manfred, characterized the end glycosylation of HER2, and based on the findings we had, he designed models in which he could manipulate genetically the phenotype of the glycosylation of this receptor. So we studied this receptor with different glycosylation features, and this is just an example of the very nice data in which that it was possible to characterize the end glycoforms by fragmentation of the egg glycan in a specific site, and this specific peptide, which includes the asparagine 530 of the receptor. So you could really have this in-depth characterization of the protein and this glycoside. But it turns out that these different glycoforms, the cells having this receptor with different glycoforms, respond differently to the therapeutic approach. So this is at different levels of analysis. So if you study metabolic activity or the cells treated with the trastuzumab, but also the activation of the receptor, and even the partners, because HER2 makes heterodimers with EGFR, and induce signaling, oncogenic signaling, and you could also observe in EGFR changes, significant changes depending on the glycosylation. It turns out that this may be mediated by different mechanisms. One could be the time of exposure of the receptor at the membrane of the cell, and the other mechanism that we are dissecting now is related to the affinity of the antibody towards the epitope in the target protein. So by SPR, you can see a clear difference in affinity of the receptor towards of the antibody towards the receptor. So by using of course we dream of applying this analytical strategy to to the clinical setting, and I hope we get there. But in an initial step, we have developed some strategies to detect in situ by proximity ligation, in this case, detecting alpha-2-6 cellulation and the protein. And some cases are completely negative for the signal, and others are clearly positive. And actually, analyzing a small cohort, we need to expand this to big cohorts. Small cohorts of this basic in situ detection in the histopathology lab, we could actually observe different prognoses of patients in the same stage, diagnosed in the same stage. So we think that the mechanism could be without treatment. The glycans are important for the signaling, oncogenic signaling, but upon treatment with the therapeutic antibody, you can have differences in response depending on the glycosylation of this receptor. And obviously these have a critical effect if you think about proper patient stratification, that because some of these patients do not respond to therapy and others do. So this could be something that we have to further implement. But the main topic I wanted to address, if I have some time, I started 5 minutes late, so I'm counting here. It's good to be a chair of your own session. So, is that the new, so glycans can also be targets for cancer therapy. And this is one of the favorites, that is the short truncated O-glycan, the CellTN, which is obviously this aberrant expression in tissues occurring in pathological situations, particularly in cancer, and actually other groups for different countries have shown that you can detect a lot of CellTN expression. It's highly frequent in cancer. And more recently, Rafael Abrantes also in my group showed that in pancreatic cancer, it's also very much expressed, particularly in the most common, the pancreatic ductal adenocarcinoma. So highly expressed in pancreatic cancer. But it's not only pancreatic, as I mentioned, and others have shown. In gastric cancer also it's highly expressed, as we observe here. So Rafaela decided to develop a new monoclonal antibody to this target. And so it was used a traditional hybridoma technology in which several clones were established, and two of the most interesting, they were screened initially against a protein rich in CellTN and a protein and this same protein lacking the cellulation removed by neuraminidase. And this initial screen showed that two clones were very reactive. No reactivity or lack of reactivity with the acylated form. And of course we used the already established antibodies that also detect CellTN, as well as the TN detecting antibody 1E3. So we analyzed one of the crucial steps when using therapeutic antibodies is to be specific to the target. And when we're dealing with glycans, we really have to make sure that you are targeting the right glycan and not presenting cross-reactivity. So we use an array of glycans, several of them containing cellulated forms and fucosylated forms. And this is a summary of the results, showing that the two selected clones were quite nicely reacting with the cellulated forms, either in threonines or serines, although one of the clones showed a cross-reactivity with acylic acid alpha-2-6 on a LAC-NAC structure. So highlighting how important it is to have a very specific antibody to the target. So of course we use these clones to characterize first in cell line models, like cell lines that were actually, some of them were engineered to display the antigen, others were naturally expressing the CLOTN antigen. So this is by flow cytometry detection of the cells by the two new clones and comparing with the established antibodies. And when we analyze the tumors, the clone that was very specific, the 52, show highly detection of the target in the cancer context. So these are tumors, we analyze different cohorts, but this is just to illustrate that they are really detecting different types of tumors: stomach, colon, pancreatic, prostate, and ovary. And when we analyze normal tissue, these antibodies did not react did not detect any of the target as expected in the normal tissue. So we look at in detail on the structure of the antibody, trying to understand where we could have hints of this defining a specificity. And this just so by multiple sequence alignment, we could compare the critical amino acids defining this specificity, comparing with the other antibodies, not only 51 and 52, but also the CC49, which is the one that have been used and implemented for some clinical detection. And there was quite a lot of overlapping in the structure, in the comparison of the structure when we align this, when we perform this modeling of the 4 antibodies. So we decided to move on to make a CAR in the hope that we could develop a new CAR T. So we use not only the 2 new monoclonal antibodies, but also the TAG72, which is described for a long time as a anti-CLOTN, and also we use a control not related CD19 that some of you may know. And we use this in different models, in cell line models also, but we move to the in vivo data, in which we analyze in different cancer models mice that are injected with the cancer cells, and we monitor the cancer cells signal upon treatment with the CAR T. So basically, summarizing, the CAR T is improving the signal, the survival of the mice, as we can see here, comparing with those immunized with the TAG72 and the mock manipulated mice models. This was done also for not only for stomach cancer, but for ovary cancer. So again, you see here the signal quite improved comparing with the other antibodies, other CARs, and also the survival of the mice comparing with the controls in these preclinical studies. Finally, we also analyze another mice model in which we xenographed patient-derived sample for mucinous colorectal tumor directly to the mouse, and the mouse was treated with intraperitoneal CAR T targeting CLOTN, and comparing the untreated with the mock treated and the 52 new CAR, we saw clear differences in delaying the increase of the tumor and also the survival of the mice in these preclinical studies. And actually, finally, we use a biobank of a patient-derived biobank of cancer patients from gastric. Here is from gastric cancer patients, in which we have the patient-derived organoids from these patients and study in the CAR T's reactivity and effectiveness against organoids with different levels of expression of the target, and actually different levels, different subtypes, histological subtypes of the tumors. So the intestinal subtype and the diffuse subtype of gastric adenocarcinomas. And we saw again from this patient-derived organoid a decrease in the viability of the tumor treated with the CAR. So with that, I finish in this very short presentation, highlighting that glycans in the oncology setting are critical for not only for diagnosis but for patient stratification to improve the capacity to define which patients will benefit from the therapies, but also they are good targets for therapies, for new therapies, and okay, this is one of example. There will be many others for sure. And with that, I finish highlighting not only the team in Porto, the University of Porto, I3S, but also the many international collaborators and national collaborators. Thank you very much.